thank you very much for that far too kind introduction, Marty. Yeah, I'm honored and humbled to be here and linked to the many fantastic scientists who have won this Young Investigator Award from ASBMB in the past and who will do so in the future, and to Mildred Cohn herself, who certainly was a pioneering scientist in so many ways. So thank you. I also am grateful for you for being here and sticking around to the last afternoon of this meeting. So today I'll be talking about my lab's work on rare earths. And if you didn't know what rare earths were, maybe the last year has informed you a little bit about what they are. You may remember that the US government has expressed some interest, let's say, in annexing Greenland and in Canada. I don't know what the status is of all those efforts, but anyway, a lot of the reasons for that are linked to rare earths. Furthermore, you may remember a number of critical minerals deals with Ukraine, with Australia, with many other countries. These are all linked to rare earths as well at the end. So why do we care about these elements? Well, they're an integral part in a number of very important technologies in a number of sectors shown here. Really, the main economic driver of their importance is their function in permanent magnets. Permanent magnets, the strongest ones are made of neodymium and iron, and terbium and dysprosium are doped in to increase the high temperature performance of these magnets in electric car, in hybrid car batteries, and wind turbines, many other things. So the reason why we are concerned about our access to rare earths is that even though the US used to be the primary producer of rare earths in the world, we ceded that role several decades ago to China, and as of a few years ago, we started mining rare earths again, but shipped them overseas for all the subsequent steps of processing up until making the magnets themselves. And the reason why we did that is that mining rare earths is really dirty, and we thought that scenes like this were untenable, and rightly so, obviously. So really what this challenge comes to here is a matter of chemistry. So the rare earths are a series of elements that are highlighted here, the 15 lanthanides plus yttrium and scandium. And I think it's instructive to just look at the somewhat funny names of many of these elements because they tell us pretty much everything we need to know about the challenges that still exist in the field in utilizing them. So here's the rare earths again. So four of them, yttrium, terbium, erbium, and ytterbium, all sound very similar. And that comes because Ytterby is a mining town from which the ores, where basically half of the rare earths were initially purified, were from. Now, Ytterby is close to Stockholm, and Stockholm is the namesake of holmium. And of course, Stockholm is in Sweden, and that's where scandium and thulium came from, so the Latin and Greek terms for Scandinavia or Sweden. Dysprosium comes from a Greek word that means hard to obtain, and that's, you can imagine why it might have been called that. And for similar reasons, we call these elements rare earths. It has nothing to do with the amount of these elements in the crust. It has everything to do about how they're purified. Now, lanthanum, the namesake of the lanthanide series, comes from a word that means to lie hidden, and that was because it was initially found as an impurity in cerium, which was the first rare earth to be purified. Now, there was an element that existed on the periodic table for a little bit, well, before it was a periodic table, that was called didymium, and that word means the twin, and that was because it co-occurred with lanthanum and cerium. And in one of the most interesting historical accidents, I think, it turned out that didymium, the twin, was actually two elements. It was praseodymium and neodymium, the green twin and the new twin. So this is all to say that the rare earths, co-occur together in the crust, and when it comes time to harvest them and turn them into technologies, the separation of one from another is extremely difficult. And that's because these elements predominate in the +3 oxidation state. And furthermore, their sizes are very similar. So if you look at the top of this slide, lanthanum through lutetium, those circles are to scale for the ionic radii of these elements. So you might be able to tell the difference between lanthanum and lutetium, but you probably have a tough time telling the difference between lanthanum and cerium. So this underlies why we continue to have difficulties in managing rare earths and purifying rare earths. And so this is an example here of how rare earths are actually purified. It goes through dozens or hundreds of steps of solvent extraction to get from the ore to a highly purified element. And when these elements are processed, it needs to be, the elements need to be like 99.99% pure at the end of everything. So this is obviously a challenge. It's really inefficient, so the process works, but it's very inefficient. It involves the use of a lot of environmentally destructive chemicals, and it costs a lot of money to do this. And so I and many other people who are working in this area now think that we have to do something fundamentally different. So you might think, who let this guy into this meeting to tell us about, about processing and mining and all that stuff? Well, it turns out that, I am really a biochemist at heart. Marty was not lying in his introduction about my background. And I became interested in this problem because I was just generally interested in metal ion selectivity in biological systems. And so for a couple years before my lab started, which was in 2016, it was really the seminal development in the field was showing that there are bacteria that utilize certain rare earths as a specific part of their metabolism. And the first function of rare earths that was shown in biology is incorporation into a methanol dehydrogenase enzyme in methylotrophic bacteria. These are fascinating bacteria. If you're unfamiliar with them, they are associated with plants, but in many cases, such as the one that's shown here, but they're also in soil, in water, and many other places in the environment. And so, as I mentioned, the key finding is that there's a particular methanol dehydrogenase enzyme. Actually, it appears to be the ancestral one, because there's also a calcium-dependent enzyme. It requires lanthanides for function. And most of these organisms prefer to use some swath of the lanthanide series that's focused at the beginning, lanthanum through neodymium, which are the most abundant elements, most abundant rare earths in the crust. So when I started my lab, I was really interested to look beyond this methanol dehydrogenase enzyme and consider how these bacteria were able to take up the rare earths and incorporate them into the enzyme, because I figured that we would probably learn something really interesting about selective recognition of these elements. And maybe if we were lucky, some of the things we found could enable a biomining approach for rare earths. And so we didn't really know what we should be looking for. So we just purified the methanol dehydrogenase from the bacterium one of the bacteria that had been shown to use these elements in the presence of lanthanides. And what we found, a good amount of the way through the purification is a number of co-purifying proteins. And we cut those out of the gel. and sent them for mass spec analysis, and one of them looked really interesting, and that was because it had 4 copies of an EF hand motif. And EF hands are very well-known calcium-binding motifs, usually calcium-binding motifs. In biology, there are hundreds of characterized EF hand proteins that bind calcium. But there were some unique features in this particular protein, including proline residues within the EF hand at that second position and an extra carboxylate that could potentially be a ligand, we thought. And so we purified this protein heterologously and started doing sort of typical EF hand protein things. And one of those was using circular dichroism to look at the conformational change that we hoped might accompany metal binding in this protein. And as you can see here, this was really the key data that showed us that something interesting was going on. This protein responds in terms of an increase in alpha helical content to all the lanthanides plus yttrium, and it also does this to scandium, but there was very little response to calcium. And to put some more quantitative information behind this finding, eventually we figured out a way to measure the apparent KD of this protein for lanthanum, and you can see that it's in the low picomolar range, whereas the protein eventually does respond to calcium, but at 10 to the 8th fold higher free concentrations. And furthermore, you can also see that the conformational change is not identical between these 2 metal-bound forms of the protein. It actually took some work with radioactivity from our collaborators using actinium as a surrogate for lanthanum. Actinium is one of the rarest elements around, but it has similar ionic radius to lanthanum. And these studies showed that the protein actually has close to a trillion-fold preference for actinium, and therefore also lanthanum over a bunch of other divalent metals that are common competitors for metal binding sites. So our general model for what's going on here, which I don't have time to go into all of the details for, is that this protein is somewhat disordered. Actually, the first LAN-M protein, lanmodulin protein, is quite disordered in the apo state. And upon lanthanide binding, it forms a compact unit that looks like this. Is from the NMR structure of the protein with yttrium bound. Whereas other elements, it's not so much that they don't bind, but when they do bind, they only partially order the protein, and that's denoted by this still a little floppy state in the bottom. And so that state is kinetically much more labile, and that's the essence of the selectivity, we believe. So we had found this protein LAN-M. We looked in the genome for where it was, and it turned out that it was in a gene cluster of 10 genes, and many of these genes looked like typical genes that would be involved in a metal uptake pathway, where a chelator was being secreted into the growth medium and taken back into the organism to mediate metal uptake. But there were a number of genes in this cluster that were very unusual, and with very little homology to known proteins. And one of those was this protein that we ended up calling LAN-D. We suspected that it would be a lanthanide binding protein as well. It looks nothing like LAN-M. It has this very weird lanthanide binding site on the surface of the protein, where only half of the ligand sphere is provided by the protein itself. But interestingly, this protein LAN-D has a very similar selectivity trend to LAN-M, at least among the beginning elements of the lanthanide series, which are the ones that the bacteria can use. And it turns out that these 2 proteins interact. LAN-D is weaker than LAN-M, and it's able to transfer lanthanide ions to LAN-M itself. And so we've proposed that this is a chaperone protein for delivering lanthanides to LAN-M in the cell. So this, of course, begs the question of what in the world are all these proteins that I've told you about actually doing? We've done all this biochemical characterization, but what's the point? And I think really the key realization is that these proteins have the opposite selectivity of the organism overall for lanthanides. So this particular organism would really prefer to use lanthanum, to incorporate lanthanum into the methanol dehydrogenase enzyme. But both LAN-D and LAN-M prefer to bind neodymium. And so what we proposed is that these proteins act as a sort of siphon. So this organism, wants to be able to use a swath, multiple different lanthanides if necessary, but it still has preferences within that group of metals. And so if there's some lanthanum around and some neodymium around, the organism will put lanthanum into the enzyme, but maybe it will have a use for neodymium at some later point, or maybe if there's no lanthanum at all, it would still prefer to put neodymium in that enzyme. So what we've proposed is that LAN-D and LAN-M are sort of a shuttle aspect of this overall uptake pathway that controls the selectivity of lanthanide uptake, because whatever the chelator is, it is not selective enough, perhaps by design, so to speak, to distinguish between all of these very similar ions outside of the cell. So there's still a lot more work to do to test aspects of this model, but this is the model that we have. So this protein, and I'll focus on LAN-M for the rest of the talk, it was really selective, and we figured maybe we can see if this can do something useful. And so the initial experiments were really exciting. The protein hangs on to lanthanides down below pH 3, and the protein itself, you can drop down to pH 2 or 1.5 or even lower, and it will spring back the next time you give it lanthanides. You can also heat the protein to above 85 degrees, which is pretty remarkable. I had worked with a lot of fragile, I should say, proteins in my time. And it was just astonishing to me that this protein was so robust. So what we envisioned is that we could essentially expose LAN-M to a solution that contained a lot of different metal ions, and then physically separate the protein and whatever was bound to it from whatever was not bound. And initially this was done with a leachate of electronic waste, which contains lanthanides, such as, your cell phone, a disembodied form of your cell phone. So it turns out that when you do this experiment, and I should note that this application work has been done in close collaboration with some tremendous collaborators at Lawrence Livermore National Lab. And so as you can see, the protein depletes the neodymium, praseodymium, and dysprosium that's present in the solution, and pretty much nothing else. So the binding was essentially quantitative, and it seemed like nothing else was binding. So then we and our collaborators immobilized the protein onto a column. This would potentially be a more scalable approach. And so, of course, we wanted to make sure that the protein was still behaving well when it was immobilized in this column format. And we also applied a much more complex solution to it. So this was a leachate of coal fly ash, which has, dozens of different elements in it. And as you can see here, most of these other elements were flowing through the column, whereas all the rare earths were binding to the column. And this enabled, upgrading a purity of this solution by about 2,000-fold, just with one passage through a column. So this first protein, it wasn't that good at separations within the lanthanide series, but it seemed to be very good at total lanthanide extraction. So we turned to analyzing what we thought was the whole family of lanmodulin proteins, and there's about 700 or so as of a few years ago that are in genomes. There are more now, and we focused on this small cluster of proteins that were very distinct, less than 30% sequence identity from the initial protein that we studied, and this is a protein that we call Hans, affectionately. We try to name all of the proteins in an anthropomorphic way. So Hans was really interesting because so even though the MEX protein, the first protein that we studied, is always a monomer in all conditions, the Hans protein is a dimer in the presence of the light lanthanides, and then as you go left to right across the series, that dimer gets weaker and weaker, and eventually it falls apart. And so why was this happening? We turned to structure, and with the help of my colleague at Penn State, Amy Bull, in her lab, we characterized the structure of this protein, and one of the metal binding sites was at the interface of the dimer here. And some of the metal binding residues were engaged in a hydrogen bonding network with an arginine residue that's conserved in this subset of lanthanides. And the structures in the presence of lanthanum and dysprosium showed us what was going on here. So with lanthanum, one of these ligands is bidentate, whereas with dysprosium, which is a little bit smaller, that carboxylate now cannot reach the metal ion unless it moves a little bit over and coordinates in a monodentate fashion. And so that messes up the hydrogen bonding network at the interface, and it starts a cascade of changes essentially that lead to the dimer falling apart. So now we were excited to use these new proteins for separations. At first not using the dimerization behavior, but more recently being able to use that dimerization for enhanced selectivity. And so we started with looking at electronic waste leachate. Again, now we can do really nice separations even with tiny columns of neodymium and dysprosium, which are important in electronic waste, as I mentioned before. And now we can begin to do much more difficult separations of metals that are more similar in ionic radius. And we have data with more complex or derived feedstocks too, but unfortunately I can't show them to you today, plus I'm out of time anyway, of course I am. So what we envision is that we can essentially replace, in certain cases, the really difficult liquid-liquid extraction part of the process of rare earth separation with these protein columns, or perhaps protein implemented in a different manner. And this technology has been, aspects of this technology have been licensed by 2 startup companies, which is very exciting. So I think biochemistry has a lot, biochemistry is obviously very important for health, right? And there are many aspects of biotechnology that utilize that importance, but it's important to remember that biochemistry is important beyond health as well. And I think there is potential here for biochemistry to change how some aspects of mining are done. So what we've shown is that biology makes some really, really neat proteins and other molecules as well that are highly selective for rare earths. These are the 2 that we've focused on mostly so far, but there are, I'm sure, many more. I think rare earth separations using proteins are a nice way of merging selectivity that comes from biology with scalability and rapid kinetics that come from chemistry. And I think it's possible that similar approaches could be used for recovery of other metals that are really important for technology, and this is an area that my lab is really interested in going forward. So I want to thank the many people who contributed to this work. My lab, of course, I showed some of the main contributors on the individual slides. I'm grateful to my collaborators, both at Penn State and Livermore, and many other institutions, so I shouldn't have said both. I'm grateful also for many funding agencies for supporting this work, and thankful for you for your attention. I'm happy to answer any questions you may have.